Unit 3 is a review of immunology and complement. It is crucial that you spend quality time with your textbook. The lectures present an overview, and the textbook has tables, charts, and illustrations which go into greater detail. Immunohematology is the study of blood group antigens and antibodies. It's closely related to the field of immunology, but the application is strictly for detecting blood group antigens on cells and blood group antibodies in the serum or plasma. Red cells, white cells, platelets, and plasma proteins can stimulate an immune response due to transfusion or pregnancy, which are the two most common methods of exposure to blood group antigens. This section provides a review of the concepts presented in the immunology course. Let's discuss or review the general properties of antigens. Antigens are large molecular weight proteins or polysaccharides which may be located on the surface or as an integral component of the cell membrane. And again, we're applying immunology to cells present in the blood. When introduced into the body, these antigens may be recognized as foreign and provoke the stimulation of immune antibodies, which will then bind to an antibody or a T-cell receptor. Antigens on allogeneic, which means the same species, cells from a donor may be considered foreign and elicit an immune response from the recipient. So genetically, we're all made up differently, and we possess different antigens inherited from our mom and dad, which are different between individual and individual. The only individual that has the same antigens are identical twins. The body's immune system normally recognizes and tolerates self or autologous antigens. Haptens are molecules of insufficient size to stimulate an immune reaction, but when they're attached to a larger carrier molecule, the body then may recognize them as foreign, and this will stimulate an immune response. A prime example of a hapten is penicillin, which we will talk about drug-induced hemolysis later on in this course. The physical nature of antigens is very, very important in determining whether or not the body will elicit an immune response. It must have molecular complexity and rigidity. Proteins are the best antigens, followed by complex carbohydrates. It has to be foreign in nature, and the greater the difference from self, the better the antigen. Molecular size, the molecular weight must be greater than 10,000 Daltons to be the best immunogen or antigen. Route of administration and dose is also very important. Intramuscular or intravenous injections are better routes for eliciting immune responses. The dose, if you get too much antigen, this may result in immune paralysis, meaning you're just overwhelmed and decide to give up. Not enough antigen may not even elicit an immune response. In speaking about blood group antigens, the chemical structures embedded in or protruding from red cells, white cells, and platelets have three common forms: glycoproteins, which are part of the HLA system; glycolipids, which is the ABH, Lewis, I, and P blood group systems; and proteins, which are the RH, M, and N blood group systems. I know this is a totally different language, but we're going to learn about the different structures present on the red cells and how immunogenic they are when exposed to them. We're going to do a brief review of the immune response. The immune response is a complex interaction between antigens and several types of leukocytes. The B lymphocytes are involved in the humoral or the antibody-producing response. Recipient B cells may recognize transfused donor red cells as foreign through these B cell receptors. This recognition causes B cells to present the antigen to a specific T lymphocyte. After presentation, the T cell cytokines signal the B cell lymphocyte to transform into plasma cells, which then produce an antibody specific for a single epitope. So, for example, when you are exposed to streptococcal organisms, your body does not make an anti-strep organism antibody. It selectively picks the best antigens or epitopes on the streptococcal organism to make antibodies to. The antibodies bind to the target antigen, which will then cause the destruction of the target cell. Memory B cells are immortal cells which, when exposed to the same antigen, immediately transform into plasma cells and produce specific antibodies to the antigens. So the plasma cells are the little antibody factories that produce massive quantities. And we'll do a review of the primary immune response when you first see the antigen versus a secondary immune response, which this is the second or more times that you've seen it. Another term for antigen is immunogen. The best antigens are proteins, carbohydrates, or lipids. Complex compounds tend to be more immunogenic than simpler molecules. T lymphocytes play an important role in regulating the production of antibodies by the B cells. T helper cells not only activate B cells to secrete antibodies, but they also activate macrophages to destroy ingested microbes, which then activate the cytotoxic T cells to kill the infected target cells. T suppressor cells close down the immune response after the invading organisms are destroyed. You do not want your immune system working overtime once the assault is over. You need to settle down. T cytotoxic cells, also known as killer T cells, kill virally infected cells and tumors. T cells have surface proteins known as cluster determinants, CDs. Helper T cells are CD4 positive cells, and they enhance and promote the action of other immune cells. Suppressor T cells are CD8 positive, and again, they suppress the immune reaction and the cytotoxic effects. 65% of your T cells are helper, 35% are suppressor, with a ratio of 2 to 1. When a person goes from being HIV positive to AIDS, that means their suppressor and helper cells have flipped, that the predominant cell is now the suppressor cell, which shuts down their immune system, which lays them wide open to a variety of different infections. This illustration shows you how the activated T cell interacts with B cells to transform them into plasma cells. Antigenic determinants or epitopes. These are structures on antigens that are recognized as foreign by the immune system. Remember, you don't just make an anti-streptococcal organism antibody, you make antibodies against specific epitopes or antigens on the microbe. An immune response is directed against specific determinants, and resultant antibodies will specifically bind to them. They won't bind to other antigens. They will bind to the antigen that elicited its response. Multivalent antigens may elicit antibodies of different specificities. And again, I bring back strep because they have this type of antigen here, this type of antigen here, this type of antigen here on the surface of the microbe. They're not all the same epitopes. They're all different. And so you may have antibody production against a variety of antigens on the surface of the organism. Unfortunately, antibodies produced in response to one antigen may cross-react with other antigens having a common determinant. And this is what people don't understand. Serological tests will always have false positives because they may react against an antigen that is very similar but not the same, or false negatives, in which that's usually due to the immune response. In blood bank, we're concerned with allogeneic antigens from other humans, and sometimes, in rare instances, autologous self-antigens. This picture represents epitopes and antibody productions. So in the yellow highlighted, you can see 4 different epitopes. And then on the right-hand side, there's the potential that this person will make antibodies to all 4 of the different epitopes. Antibodies are a family of proteins called immunoglobulins, and we usually abbreviate it IG for short. They're structurally related with 2 common functions. They combine with antigen, and they mediate various biological effects. Antigens are glycoproteins composed of 4 polypeptide chains joined together by disulfide bonds. The terms antibody and immunoglobulin are used interchangeably. Antibody is just a lot easier to say. There's 5 classifications of antibodies, which are listed below, and we will cover in more detail later. The 5 classes of antibodies are differentiated based on certain physical, chemical, and biological characteristics. Each antibody molecule has 2 identical heavy chains and 2 identical light chains, either kappa or lambda, which are joined by disulfide bond bridges to form a Y-shaped molecule. The bridge provides flexibility to the molecule to change into a 3-dimensional shape. So I want to grab the antigen over here, and I want my other antigen binding site to be able to stretch over here or stretch backwards. So again, flexibility to bind to more than one antigen. Each heavy chain has characteristic features which permit them to have unique biological properties. Due to its secretory component, IgA is the only antibody capable of residing in mucosal linings. IgE activates mast cells, causing hypersensitivity reactions. IgD is an antigen receptor on the naive B cell. Still not much known about it. And IgG and IgM are the antibody classes most important to transfusion medicine. And again, there's 2 types of light chains, kappa and lambda, which are identical on the antibody molecule. You can't have a kappa and a lambda on the same antibody molecule. Impossible. Again, we're dredging information to the front of your brain, and we're going to review the 2 types of immune responses. There's the innate or nonspecific immune response, and there's the adaptive or specific immune response. Innate immunity involves the body's first line of defense. These include physical barriers, which include intact skin and mucous membranes. If, let's say that you get a puncture wound and that physical barrier is breached, then an infection could potentially set up. There's also physiological factors, hydrochloric acid in your stomach, ciliated epithelium in your respiratory tract. These are little hair-like fibers that the microbes will get stuck on, and then they're wafted back to the top of the respiratory tract where they're expelled. Flushing action of urine, unsaturated fatty acids on skin make it very uncomfortable for pathogens to set up housekeeping. Sweat and tears because they have a high salt concentration, which is detrimental to the life of bacteria. And commensal normal flora, especially on your skin, you are covered with bacteria. But this is good because it prevents the pathogens from being able to set up an infection on your skin. Inflammation is the body's reaction to injury, and it's known as the body's second line of defense. And this results in increased blood supply to the area, increased capillary permeability, which allows the migration of leukocytes into the surrounding tissues. These 3 events manifest symptoms which include pain, heat, redness, and swelling. I still love this picture. I want you to spend some time with it so that you can understand what the process is in inflammation. So we have the first and second lines of defense, and then we also have cellular immunity. These are important defense mechanisms against viral infections, some fungal infections, parasitic disease, and against some bacteria, particularly those inside cells. They're also responsible for delayed hypersensitivity reactions, transplant rejection, and possibly tumor surveillance. T cells involved are the T helper and the T suppressor, which regulate the intensity of the immune response, which we just discussed. Please review your immunology notes from the fall for more specific information. Lymphokines are chemicals involved in the immune response. They attract neutrophils and monocytes to the site of infection. They cause aggregation of macrophages at the site of infection, and then they activate macrophages to phagocytose and destroy the invading microorganism. The combined result of lymphokines is amplification of the inflammatory reaction. Macrophages are also important as part of the humoral immune response. They have receptors for the Fc, the fragment crystalline portion of the immunoglobulin molecule. Antigens are coated with antibody, will be bound to the macrophages. So this causes removal of these antibody-coated microorganisms through phagocytosis, where they're engulfed and destroyed. Macrophages have receptors for complement component C3b. Antigens coated with C3b will bind to macrophages. Some blood group antibodies, especially ABO, are capable of activating complement. This will result in destruction of the red cells, also known as cytolysis. Involved in cellular immunity and is very important in rejection of transplanted tissues. Macrophages participate in phagocytosis, inflammation, and cellular immunity. Macrophages are mainly involved in nonspecific immunity and include the phagocytic cells, your mononuclear phagocytes, your polymorphonuclear phagocytes, which are your neutrophils, eosinophils, as well as mediator cells, which are your basophils, your mast cells, and your platelets. Adaptive immunity is also known as specific immunity and humoral immunity. This involves the ability to recognize self from non-self. Encounters with non-self or foreign materials results in the production of antibodies, the humoral immunity, or actions of T cells in the cell-mediated immunity. Immunohematology is primarily concerned with the causes and effects of humoral immunity. The humoral immune response involves the productions of antibodies induced when the host immune system encounters foreign antigenic substance and reacts to this antigenic stimulation. There's two types of responses, primary and secondary. Secondary is also known as an anamnestic response, and you must know these two responses from beginning to end because these are the backbone of blood bank. This is mediated by B lymphocytes, which are then transformed into plasma cells. In the primary immune response, the antigen is ingested by an antigen processing cell, an APC. This is presented to the helper T cell, which has receptors for the antigen. The activated helper T cell presents the antigen to the B cells and secrete lymphokines, which activates the B cells to produce antibodies. The B cells proliferate and differentiate into plasma cells, which secrete the antibody, as well as memory cells. Initially, there's low affinity and avidity. Affinity is how much I like you. This gauges the best fit between the antigen and the antibody. The most common example is the lock and key fit. Avidity is the strength of the reaction that keeps the antigen and the antibody together. The higher the avidity, the more likely the antibody will stay stuck to the antigen. So it's very important. Initially, there's low affinity and avidity, but as time progresses, they kind of fine-tune the production line to where these become very high. IgM antibodies are produced first, followed by IgG. The primary immune response takes weeks to months. So once you're exposed to an antigen, it takes a while for the mechanism and the machinery involved in the humoral immune response to get up and running and then become very efficient and effective. The secondary immune response results due to exposure to the same antigen again. This activates your B memory cells who know how to do the job, and they proliferate into plasma cells. This causes the production of IgG antibodies, which have high affinity and high avidity. Some production of IgM may occur due to the immune cells which have not seen this antigen before. So they're going to go through a primary immune response. But IgG is the predominant antibody produced. So we get rapid production with a high titer. This takes hours to days. So we get an immediate response to this second exposure. Antibody production in the humoral immune response occurs in 4 phases following the antigenic challenge. The lag phase, when no antibody is detectable. The log phase, in which the antibody titer rises logarithmically. The plateau phase, during which the antibody titer remains steady. And then the decline phase, during which antibody levels gradually decline. Because once the invading microbe is destroyed, then there's no need to have these very high levels of antibody. You must be able to differentiate a primary versus secondary immune response based on the following: the time, the antibody titer, the antibody class involved, and the antibody affinity and avidity. I would recommend making a little chart with these on one side and then IgG and IgM on the others. These are critical to understanding reactions obtained in blood banking. The following chart nicely illustrates the concept. Once again, you need to memorize the specifics that define a primary versus a secondary immune response. This is the end of part 1. Again, be sure to review your objectives, your textbook, and this presentation. Write down any questions that you may have going to class.